Welcome to the fascinating world of living droplets and chemical oscillators, where chemistry meets the emergence of lifelike behavior. These droplets oscillate in color, pulsate rhythmically, and communicate with neighbors through chemical waves, displaying behaviors remarkably similar to living cells. Let's start by understanding what chemical oscillators are and why they're so remarkable. Chemical oscillators are reactions where the concentrations of reactants and products change periodically over time, like a chemical heartbeat. Unlike normal chemical reactions that settle into equilibrium, oscillators require constant energy input and remain far from equilibrium, just like living systems. Here we see how concentrations oscillate over time, creating beautiful periodic patterns that never settle down to a constant value. The most famous chemical oscillator is the Belousov-Zhabotinsky reaction, discovered in 1951. Boris Belousov discovered this remarkable reaction while trying to model the Krebs cycle, a fundamental metabolic process in living cells. The reaction involves cerium ions, malonic acid, and bromate ions in an acidic solution, producing stunning color oscillations. The solution oscillates between red and blue colors as cerium ions switch between oxidation states, creating a mesmerizing visual display. Here's an interesting question. Why was Belousov's discovery initially rejected by the scientific community? The answer is that it seemed to violate the second law of thermodynamics as it was understood then. Scientists thought spontaneous oscillations were impossible in closed chemical systems. Now let's dive into the mathematics that governs these oscillating systems. The oreganator model, developed in the 1970s, provides a simplified mathematical description of the Belousov-Zhabotinsky reaction kinetics. This differential equation describes how the concentration of bromous acid, represented by X, changes over time with 4 key rate constants. Similarly, this equation describes the bromide ion concentration Y, which acts as an inhibitor in the system. And this third equation tracks the oxidized catalyst Z, completing our coupled system of nonlinear differential equations. These coupled equations create what mathematicians call a limit cycle in phase space, a closed trajectory that the system repeatedly follows. In phase space, we see the system tracing out an elliptical orbit, returning to the same states periodically, regardless of initial conditions. When we add spatial dimensions, chemical oscillators create even more remarkable patterns. In a petri dish, the BZ reaction creates traveling waves that propagate across the medium like ripples on a pond. The reaction-diffusion equation combines diffusion, represented by the Laplacian operator, with the reaction kinetics function F. Chemical waves emanate from oscillating centers, spreading outward in concentric rings that can travel several centimeters across a dish. These waves travel at typical speeds of 1 to 10 millimeters per minute, slow enough to watch with the naked eye. Now let's explore what happens when we confine these oscillating reactions inside tiny droplets. When we create droplets of BZ solution in oil, each droplet acts like an individual chemical cell with a membrane boundary. Each droplet contains the oscillating chemistry, but they're physically separated by the oil phase, creating isolated oscillators. These droplets are typically 50 to 500 micrometers in diameter, about the size of human cells or small sand grains. With a radius of 100 micrometers, a droplet volume is about 4 times 10 to the minus 9 liters, containing billions of molecules. Here's where things get truly fascinating. Droplets can synchronize their oscillations like fireflies flashing in unison. Let's ask a crucial question. How can physically separated droplets synchronize their chemical oscillations? The answer is through diffusion of inhibitor molecules through the surrounding medium, creating a communication channel between droplets. Droplets exchange inhibitor molecules that diffuse through the oil phase, creating weak coupling that can lead to synchronized behavior. This is the Kuramoto model for coupled oscillators, where theta represents the phase of each oscillator and K is the coupling strength. The Kuramoto model predicts that above a critical coupling strength, oscillators spontaneously synchronize into phase-lock states. Droplet networks can exhibit several types of synchronization patterns. In-phase synchronization occurs when all droplets reach their maximum concentration at the same time, like a chorus singing in unison. Here, all droplets are in the same red state simultaneously, perfectly synchronized with zero phase difference. Antiphase synchronization creates alternating patterns where neighbors oscillate out of phase by 180 degrees. Neighboring droplets show opposite colors, creating a checkerboard pattern of oscillations. Traveling phase waves create sequential activation patterns, where the oscillation propagates along the droplet chain, like a Mexican wave. To quantify synchronization, physicists use a mathematical tool called the order parameter. The complex order parameter, Re to the I psi, is the average of all phase vectors, where R measures synchronization strength. When R equals 0, the oscillators are completely desynchronized. When R equals 1, we have perfect synchronization. The order parameter R increases sharply at a critical coupling strength K, marking the transition to synchronized behavior. Beyond the BZ reaction, enzymatic systems also create oscillating droplets with biological relevance. Glycolytic oscillations occur naturally in yeast cells during sugar metabolism, showing that living systems use chemical oscillators. During glycolysis, ATP concentration oscillates periodically, creating an energy rhythm in the cell. Enzymes act as catalysts that create feedback loops, essential for generating sustained oscillations in biochemical networks. Enzymatic oscillators typically have periods of 1 to 10 minutes, much slower than BZ reactions which oscillate in seconds. The diffusion field creates an invisible communication network between droplets. Fick's first law states that the diffusion flux J is proportional to the concentration gradient, with D being the diffusion coefficient. For small inhibitor molecules in aqueous solution, the diffusion coefficient is typically around 10 to the minus 9 square meters per second. The diffusion timescale is approximately L squared over D, where L is the characteristic distance between droplets. If droplets are separated by 1 millimeter, it takes about 15 minutes for inhibitor molecules to diffuse between them. Chemical signals spread outward from each droplet in diffusion halos, overlapping to create coupling between distant oscillators. When many droplets interact, truly emergent collective behaviors arise that no single droplet possesses. Let's consider an important question. What makes this behavior truly emergent, rather than just complicated? The answer is that global synchronization patterns emerge from purely local interactions, with no central coordinator. Each droplet only knows about its immediate neighbors through diffusion, yet the entire network can synchronize into coherent patterns. This is similar to how heart pacemaker cells, neurons, and fireflies all synchronize through local interactions to create global rhythms. In some conditions, droplet networks form chimera states, where order and disorder coexist. Chimera states, discovered in 2002, are fascinating patterns where some droplets synchronize, while others remain chaotic, all in the same system. In a chimera state, one group of droplets beats in perfect synchrony, while another group oscillates chaotically, defying intuition. Chimera states require non-local coupling, where each oscillator couples not just to neighbors, but to distant oscillators as well. Some chemical droplets can even move themselves, showing primitive forms of locomotion. Chemical gradients on the droplet surface create Marangoni flows, surface tension gradients that can propel droplets like tiny chemical motors. The propulsion velocity is roughly the surface tension gradient divided by viscosity, giving speeds around 1 millimeter per second. The droplet pushes fluid backward on one side, propelling itself forward like a microscopic jet engine. Remarkably, these self-propelled droplets can chase each other, avoid obstacles, or form trains of multiple droplets moving together. Modern computers allow us to simulate these complex systems and predict their behavior. Finite difference methods discretize space and time to numerically solve the coupled reaction-diffusion equations governing droplet behavior. This finite difference scheme updates the concentration at each grid point based on diffusion from neighbors and local reaction kinetics. A typical simulation might use a 100 by 100 spatial grid with time steps of 0.01 seconds to capture the dynamics accurately. Depending on system size and duration, these simulations can take minutes to hours on modern computers to reveal emergent patterns. Droplet networks can perform primitive forms of information processing and computation. By arranging droplets in specific geometries, researchers have created AND and OR logic gates, where droplet states represent binary information. Two input droplets converge on an output droplet, where the combined chemical signals determine whether the output oscillates. Droplet arrays can recognize spatial patterns by their collective response, similar to how neural networks process images. These chemical systems offer insights into how life might have emerged from non-living chemistry. Chemical droplets exhibit lifelike properties, including metabolic cycles, self-organization into patterns, and chemical signaling between units. Chemical oscillators may represent an intermediate stage between simple chemistry and primitive living cells called protocells. Here's a profound question. What's still missing from these chemical droplets to be considered truly alive? The answer is heredity, evolution, and true reproduction. These droplets copy their behavior, but not their chemical instructions. Let's explore how scientists actually create and study these remarkable droplets in the laboratory. Microfluidic devices with channels smaller than a human hair create uniform droplets by breaking up fluid streams at precise junctions. These devices can generate 100 to 1,000 identical droplets per second, creating populations for statistical analysis. In a microfluidic channel, droplets flow like beads on a string, each containing the oscillating chemistry isolated by the carrier fluid. Fluorescence microscopy with high-speed cameras tracks chemical concentrations inside droplets in real time with millisecond resolution. These systems require constant energy input to maintain their far-from-equilibrium dynamics. The second law of thermodynamics still applies. Total entropy increases, but the system exports entropy to maintain internal order. Each oscillating droplet dissipates approximately 1 nanowatt of power, about a billion times less than a typical LED light. This power comes from the chemical potential energy of reactants, which is converted to heat during the oscillation cycle. Chemical oscillators and droplet networks have exciting potential applications in various fields. Smart materials incorporating oscillating droplets could sense environmental changes and respond autonomously without electronics. Drug delivery systems using oscillating chemistry could release medications in pulses, matching the body's natural biological rhythms. Self-propelled droplets could be used for targeted delivery of therapeutic agents to specific locations within the body. In conclusion, chemical oscillators and droplet networks exhibit remarkable properties that can inspire new technologies and help us better understand the emergence of life.